So antiprotozoal. Now neutrophil. This is for antibacterial. So how we do it? We can do an easy test for that. If you have a cold-like symptoms, you go to see a doctor. The normally routinely operation in the lab is they give you a swab to touch your nose, then to see a rapid testing whether you have Streptococcus pneumoniae. If you have a Streptococcus pneumoniae, then they give you antibiotics immediately. Let's say penicillin, amoxicillin. But there's another way what we can do, we don't often do here in the United States, in the other country, we get your blood. We get your blood cell. The blood cell, we try to see your white blood cell total population. If you see the number is more than 10 million, absolutely you have an inflammation. Then they go to see the basophil, eosinophil, and the neutrophil. If they find your neutrophil is above the normal range, then we can say you have a bacterial infection. Because neutrophil, if it's activated in your body, which means it started antibacterial process, which means you have a bacterial infection. So that's another way we can check whether your cold-like symptoms is caused by virus or caused by bacteria. If it's caused by bacteria, you have to use antibiotics, but if it's not, you shouldn't do it because it will create antibiotic-resistant bacterial community. So that's a tricky thing for that. Okay, so this is the one we finished talking about that. We will go into a very routine one, which is we call it immunity. So what is the real immunity? What we talk about is humoral immune and cell-mediated immune. Humoral immune, you will generate a B cell, and cell-mediated immune, this is actually should call T cell mediated immune, but people think you're smart, so they took this out. Then we'll activate T cell. Now the major difference between the B cell and the T cell, it is not the memorization. The B cell, T cell, they all have a memorization. Obviously, B cell will be a little bit longer time. The major difference is B cell will generate antibody and T cell will not. Now, where the B cell comes out? I cannot draw the picture very well. What happened is you have a chicken, let's say, this area. You have a chicken. The first time people find it, it's a long time ago, about like 150 years ago. In the back, in the butt area, we also say the muscle surrounded with chicken asshole. And they will have a very immune-activated tissue area. They generate this specific area. We call it bursa fabricis. And this bursa fabricis have lots of the immune-activated cell. So, and created by a scientist called Fabricus last name. So then we want to memorize that, and we shorten the whole words and become B cell. So that's what the B cell originally comes from. And B cell obviously is coming from the colonization process. It will generate lots of the ideally exactly the same the cells we call it a B cell. Okay, so this is some of the overall pictures. So let's go back to see the slides. What we talk about that? So this slides we talk about the host-resistant overview. The most pathogen disease-causing microbes. Now they overcome surface barriers which underlie. That's what we mentioned about need to come back with the salts, with the mucous membrane, all those systems. Then the body will generate a non-specific resistant and a specific immune resistant. And we mentioned this is non-specific. This is also non-specific, more universal. This is specific. And then we have some terminology here. So what is called the immune system? It's composed by the widely distributed cells, tissues, organs, recognize a foreign substance, neutralize, destroy them. That's what we already talked about. Immunity, capability for the host to resist a particular disease. Immunology, it's a science behind it. And then we already spent time talking about non-specific, specific. So this is slides we're going to pass it, and this is one we already gave you some examples. The first surface defense system have a mechanical, chemical, microbial, phagocytic defense system, and immunity, humoral and cell-mediated immune. And this slide is more clear, showing you the picture, what we talk about here. You can see the conjunctiva, lysozyme, flushing of the tears, respiratory tract mucus, normal intestinal flora, flushing the urine sample, oral mucus, alimentary tract mucus, stomach gases, and the skin surface. This is the one we already taught. And I think this is an excellent slide which tells you the normal human microbiota in our body. Lots of them we already mentioned. For example, nasal area, you see Haemophilus influenza, Streptococcus pneumoniae, and corynebacteria. And we even did a throat culture in the lab. Lots of you find corynebacteria. Then the mouse, we have a Viridans Streptococcus, but besides that, we have others, skin surfaces. Vagina area basically will be dominant with lactobacillus. That's why the pH is usually low, and that's a defense system for the bacteria. And the streptococcus. And we also say that the vagina area has to keep a little bit wet, is not be completely dried out. If it's completely dried out, staphylococcus will be dominant. If it's end up with odorous, then you have a toxic shock syndrome, and we mentioned it. And urethral, throat culture, stomach, you didn't see lots of the bacteria. But we mentioned one special bacteria in the lab is HP. It's Helicobacter pylori, because they have a urease. So create urea, and then break down, become ammonia and carbon dioxide, and that's called the urea test. That's why they can survive, because they buffer in the pH at the stomach from 1 to 2 back to 3 to 4. Intestinal area, relatively less. We have enterococcal, lactobacillus, Candida albicans. This is a very toxic fungi. But the large intestinal area, we said the total population is more than 10 to the power of 12. And that's all type of the bacteria could be there. So you can see our whole body is composed by the human microbiota. And in our first slide, it says the microbiota, the number is at least 10 times the whole human cells. So this slide is really good. It tells you how huge population and how diversified the human microbiota in our body. And this is we already mentioned. The blood cell coming from stem cell, the myeloid, then we'll have a different blood cell. So we have a red blood cell. The scientific name we call it erythrocyte. The erythrocyte. That will be carrying oxygen. Then platelets, that's cause blood clotting. So if the patient who has leukemia, then the blood cannot be clotting lots of the time. The macrophage, this we were going to mention real quick. But we mentioned about here, for the major defense system from the white blood cell is belonging to the granulococcytes, which is neutrophil, eosinophil, and the basophil. And then you have another separate line coming from lymphoid, that's for the B cell and T cell. We're going to talk about this a little bit more. We have one thing we haven't mentioned about is phagocytosis. So this is a very easy to understand is before the body going into the real immune defense system, they will be activated in the white blood cell. Then one of the white blood cells, which is called macrophage. This macrophage let's say you have a bacteria here. This macrophage will engulf it, which is pack it. Once you pack it, it will make sure it is being surrounded with macrophage. And then they will be activate lysozyme. And the lysozyme will go through lytic process. And this lytic process we already mentioned about here is to break down the 1,4-glycosidic bonds of the bacterial cell wall system. And the whole thing, where you have here, this one, we call it phagosome. Okay, so it's very easy to understand. You develop a phagosome, then the lysosome has been activated and break down the glycosidic bonds of the bacterial cell wall, and then you destroy it. And this macrophage can be activated independently. Also, they could be activated with antibody and antigen. That gives you another terminology we call it opsonization. We will mention this later on. Okay, so next couple of slides we have here, we're going to talk about. This comes out with another important topic. We will be briefly introduce you here, which is how we develop our active immune system. So we want to talk about that, the active immune system development. And this will be a question in the final exam because it is important. So we call it acquired immune system development. So there are two levels. From a big picture, everything comes from bone marrow. This is the center. That is the reason if people have leukemia, then the best treatment is not chemotherapy, is you got to be transplant a new bone marrow. And we also mentioned in the class, there is the first broad stream antibiotics can be very effective, but they have a very severe side effect is destroy the bone marrow. Once you destroy the bone marrow, you acquire the immune system development is basically will be failed. So that is important. The bone marrow will have the stem cell. And the stem cell will go into different area. They could go thymus gland and then go to the spleen. So they go to the spleen and then go to the lymph nodes. And then they will generate this B cell. We call it a B cell line. The stem cell, they could also go to thymus gland. Of course, partially could be with lymph nodes. Then we'll generate a T cell line. That's the overall picture. But the development, it is also depends on the antigen. So let's say you have an antigen, which is a small, soluble antigen. If it's a small soluble antigen, then they go into, or we say small free soluble antigen. When they go into the body, they will directly find is a B cell. Then will generate all kinds of things like this. That's antibody. In other words, we also call it immune globulin. And then they will go through a multiple cloning process and generate more and more B cells. Each will carry a multiple antibody. So that's one line, which is related to the B cell line. Now, what's a T cell line comes out? If instead of you have a small free soluble antigen, you have relatively large complex antigen, what are you going to do? Then you go into the body, you will find something like this. Clove leaves, you can say, and then they go in here. What is this called? Dendritic cell. Or we say antigen presenting cell. So they present the antigen, and then they go there. Now they just go there. They don't not just go there naturally. They have to be recognized by something. I don't have a colored chalk pen today. These things are important. Eventually, they will be connected. What are these things? MHC. Major histocompatibility complex. This is a very important structure to recognize the foreigner. We know about that HIV patient, we call it an immune deficiency patient. The immune system is deficiency. Why? Because their MHC is damaged, cannot recognize the foreigner. And this can recognize the foreigner, which is large complex antigen. Then they will generate. We call it naive T cell. Then the naive T cell later on will develop to become T killer cell, T helper cell, and lots of the others. So this is the T cell line concept. Now, what is the major difference between B cell and T cell? Is the memorization. It's antibody. But there's another difference, is the memorization time. The T cell line is very short and the B cell line is longer. This is the reason DTaP, remember diphtheria, tetanus, pertussis, the shots you need to do is regular scheduled boosters. So what's the reason for that? This immune vaccine is only activate T cell line. They are not activate B cell line. So they have a memorization time, but it's relatively short. That's why the young kids, 2 months, 4 months, 6 months, 1 year, 15 months, 2 year, 6-year-old, later on you have to do the booster shots. So this is very important. We talk about the acquired immune system development. Now, related to this topic, we can categorize our immune development into 4 different categories. It's very interesting, we can do this conclusion. The first category, we call it natural acquired active immune. What is that? A good example is you recovered from a disease, a bacteria-caused disease. So for example, if you recover from Streptococcus pneumoniae, you have a Streptococcus pneumoniae antibody, that is natural acquired active immune. The second one is natural acquired passive immune. Where it is? What is example? Placenta. Placenta go to newborn baby. That is why usually as a newborn baby in the first 9 months, they will be healthy. They will not have any disease because the IgG could get in from the mom. Now obviously that could come from the breastfeeding also. That's another story. So natural acquired passive immune. Since we have a natural acquired active passive immune, we obviously have another two categories. Is artificial acquired active immune. What is this category? Everybody knows vaccination. You go ahead to take a vaccine of flu or COVID-19 or combined boosters, we call it vaccination. That's artificial acquired active immune. The last one, we have artificial acquired passive immune. What is a good example of this one? Anti-equine botulism. Remember, when we have Clostridium botulism, I said the first thing what you do is not treat antibiotics. Is you're going to call the CDC. Then the CDC will tell Atlanta Airport to shipping anti-equine antitoxin. That is artificial acquired passive immune. Why we say it's passive? Because it's not a prevention, it's a treatment. Okay? So that is very important. Have 4 different categories. So when we talk about here, you can go back to see the slides. We talk about types of specific immune. This is, you see the bone marrow system, the thymus gland, lymph nodes. In our body you have a spleen, lymph nodes, thymus, tonsil area. It's a major our immune system. And we talk about the B cell line, T cell line, and it's more detailed when I draw the picture there, how we developed it. So we're going to move on to the next topic. Is antigen and antibody. What are they? So obviously antigen is a substance stimulates the immune response. So give example, protein, polysaccharides, nucleic acids. The larger molecules, the better, must be recognized as a foreigner. Then they have a place to be recognized by antibody. We call it the epitope. We call it the antigenic determinants. The small sites on the antigen determine the specificity of the immune response. So the question is, what is antigen? This is what we want to talk in this class. Some concepts we want to be mention about. It's a little bit different. So we talk about the immune system. We talk about antigen-antibody reaction. So we want to talk about antigen. So what is antigen? You have a different definition, but we said it very simple in the microbiology area. A whole bacterial cell is not antigen. Antigen is their specific location. It could be a protein, most likely. It could be flagella. It could be their sugar, carbohydrates. But the whole cell is not. A very easy example. E. coli 0157H7. We mention about this all the time because Shigeru toxin, zero tolerance in non-intact beef, including ground beef. What is O means? This is O antigen. It's coming from lipopolysaccharide side chain. It's a modified sugar, we call it ceramic. What is H? It's coming from flagella. And their serological typing number is 157 and 7. That is like you gave them an ID, like they give you a WVU ID. It's the same thing. And we also talk